glucose and fructose. Exact same formula. So, same molecule, right? Nope. Two completely different sugars. And that gut feeling, same formula, same thing, is the most reasonable sounding mistake in all of Unit 1. So let's kill it, because one idea runs this whole topic. Structure decides function. Start with one monomer. Notice it has a point on one end and a notch on the other. That shape matters. Monomers only link one way, head to tail, which leaves two different ends. A free amino group here and a free carboxyl over there. One's basic, one's acidic. So the chain has a direction. We read it N to C. And the cell always adds to the same end. So a protein is born N-terminus first. DNA runs the identical argument, just with different parts. Those prime marks, they count carbons on the sugar, nothing to do with the bases themselves. Phosphate on one end, a free hydroxyl on the other. New units attach only to that end, so strands grow 5' to 3'. Now here's the payoff. Take this little peptide, gly, then ALA. Reverse it to allogly. Same two amino acids, and a different molecule. In DNA, it's even louder. ATG is a start codon. GTA isn't. Same three letters, opposite meaning. And you can't cheat by flipping a monomer around. Turn one backwards and there's just no bond to make. It even shapes the double helix. The second strand runs the opposite way, anti-parallel. So writing the partner takes two moves. Complement each base, then flip the whole thing around. Skip either move, and you've written a different molecule. You need both. Okay, zoom all the way into one monomer. This is glucose. Its carbonyl sits at carbon 1. And this is fructose. Same formula, but the carbonyl slid over by one carbon. That one shift makes glucose an aldose and fructose a ketose. Different sweetness, different metabolism. Add galactose, also C6H12O6, and you've got one formula, 3 sugars, 3 jobs. But here's the example that ends the argument. 2 fatty acids, same formula, 1 double bond each. In the cis form, the tail kinks. Kinked tails can't stack, so it stays liquid. That's olive oil. Flip that one bond to trans, and the tail goes straight, packs tight, and turns solid. A trans fat. Same atoms, 30 degrees apart in melting point. The kink is the entire difference. So unsaturated doesn't mean liquid, and it's never about mass. Isomers weigh exactly the same. One last costume. Same carbon skeleton, clip on a carboxyl, and it's acidic. Swap in a hydroxyl instead, and it's polar, but not acidic. The functional group is where the chemistry lives. So, same formula isn't the same molecule. Direction is information. Cis kinks, trans packs. Because the formula is just the receipt. The arrangement is the molecule. Want to step through it yourself? Links below.